can understand as a question for aging, right? Like, why is it so different? Is it purely socioeconomic, or, you know, I don't know, it's just some thought.
I think that part is pretty clear.
Yeah.
That's exactly what. it
Maybe,
is.
right?
Yeah.
Anyway. Okay. We had a good first round, I thought.
Everyone wants it to be like the secret seaweed, that if you just eat like one thing in the Okinawa diet, then you're fine.
Just go there every couple of years, eat on the interesting point. Oh, just to kid around a little bit, one could make an economic, like a tourist destination longevity out of that
Mhm.
Oh, yeah.
state.
Longevity tourism.
They might push their wine a little bit on that too.
There's actually a new series on Netflix I read about blue zones. It's I think it's
yeah.
I think it's a 4-part series. That I think goes around to these different areas and then asks what's different.
Somebody else mentioned that too, yeah.
So I haven't seen, I think I downloaded it, no? Need to look at that.
Should write this down.
Okay.
So
Are you-
I'm the moderator.
Okay, super.
You guys have to speak up a little bit. The microphone can pick you up, but it's in the middle. Yep.
Sure, okay.
And then the stated prompt for this one is connections between research questions and approaches that take us whatever direction your conversation takes you.
Mhm.
Does anyone want to volunteer to write and report back?
We do need someone to volunteer for that.
Mhm.
I can do.
Oh, thank you, appreciate that. It's great.
So we,
wanna
and
do
that
the
now
chat?
that we have organizers here. In our last one, we did it on our computer.
That's fine.
Yeah, we actually asked.
Because we're not gonna be pasting
Right.
and doing like dot exercises or anything like that.
No,
on this, yeah.
it's just to make it low
Okay.
energy, so whatever
Yeah.
is easiest.
This
for
is the lowest energy and then also I
Okay,
can engage. with
sounds
that.
good. Yeah, we just wanted to
Mhm.
provide the tools if somebody wanted to use the boards, right? So,
The
yeah.
other thing we found it useful to do was we like looked occasionally at what was up there.
Yeah.
And be like to make sure, so if we just like, so I got da 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 da. that
Yeah.
might- But you'll probably do it.
Yeah.
Right,
Yeah.
okay,
got that.
yeah, okay, good. Yep. Yeah, I mean, in your case, Josh, right, you seem to have so you recognize molecules also, right? That then start triggering something, right? Is that right?
Yeah.
Yes.
perfect. We recognize more. Yeah.
Your systems.
Right, some of the technologies we built, which actually Nicole invented.
Oh.
Her time as a graduate student.
That's wonderful.
First version are designed at this kind of immune motivated axis where we're trying to, for example, we knew certain cytokines are enriched in the tumor microenvironment. We wanted immune cells to turn on our cell therapies when they were in a specific combination of factors. And so we used engineered, we tried using natural receptors coupled to downstream pathways, and crosstalk limitations of the way the natural systems were precluded that from working. So then we pivoted by necessity to synthetic receptors. which do that. So in this case, they're sensing natural features of the environment. And then they relay that in a more or less orthogonal way to something inside the cell, which could be like of the signaling pathway or genetically or epigenetically. And so that's been one of the approaches we've taken to kind of make these tumor microenvironments reactive / specific cell functions.
Cool. So, I mean, if we had this aging signature that aging cells release, could that work a similar way then?
Yeah.
potentially?
Yeah, and I don't think in any way that's limited to my group. I think that's in general, I would say, a way we oftentimes think about these like input output functions. Or using the cell as the prosthetic feedback controller. That's one way of talking about it. If you wanted to modify the aging environment. So when we compare it to like a CAR T cell therapy, which is a very different type of immune therapy, we were doing this more feedback of the environment in the sense that we sense immunosuppressive cues and deliver specific immunostimulatory cues. So the engineered cell in that context isn't doing the killing.
No,
It's actually
it just,
making cold tumors hot
right.
or reactivating local immune responses. So you could flip that and use that for treating inflammatory disease for sure. People, Mark Fussenegger and a few other folks have shown the potential to do something like that. But I think the generic description of all those could be something like sensing an environmental feature and delivering a regulatory cue to try to shift that network state.
What's the delivery vehicle for that? Are
I
those
think
cells?
Yeah, so right now, I think, so we definitely are using cells. I think the the versions that are more in the biomaterials front might use, have some promise too, but they're to date limited to things like pH or matrix metalloproteinases, things that can cause a material shift. So I think the cells are harder to make and deliver, but it's easier in some ways to plug it into different aspects of biology.
So this is a big theme of our last thing is what does delivery look like? And just using cancer as a model.
Right.
Seems like there's two ways. You like program the bone marrow or something, and then these cells are going out and finding the cancer, or you're injecting it into a tumor. So
Yeah.
what is the mode of delivery that of a cell?
I think in general, you can think of it either way. There's one layer past that, which is, are you taking the cells of the patient, engineering them, putting them back in,
Sure.
or you're delivering a vehicle that then gets to the cells? And certainly in the cancer field, active work on both modalities, because even though the latter one sounds better, there's a huge safety benefit to the former.
Mhm.
So I think if you want to make it as safe as possible and as accessible as possible, the ex vivo modification approach, which is what we've done so far in most patients,
Yeah.
has a lot of advantages.
Because one of the challenges, so I think for this Senoloic approach, I think that's where you were going.
Mhm.
That makes a lot of sense. One of the questions in aging more globally is, but and specifically with synthetic biology is, what is the mapping
Totally.
like?
Mhm.
What, how do you,
Mhm.
how do you actually end up? So, and we'll get to this in a second. I know I can think of all these tool-based things for discovery
Right.
that are
Mhm.
going to be very useful.
Mhm.
And I think to be more aggressive, the question is eventual translation into therapeutics. What is that? We're not going to, you and I already talked about this. So you genetically engineer a human, but I do think introducing engineered cells.
Yeah.
Mhm.
that's,
I think it's a deployment question too, because even if you could engineer the cells, that depending on the application, you and I were talking about maybe in the brain, there'd be some attractiveness to a cell therapy because you wouldn't engineer the neurons, you'd engineer something that maybe goes through. Then even if you can engineer, let's say there's a great stem cell derived stem cell derived microglia
Mhm.
product, you still have to somehow get those microglia to migrate to where they need to be
Mm.
to do their job within that. So I think the general challenge of tissue homing or tissue distribution of an engineered cell is a big challenge. And, in some ways, cancer is a great low-hanging fruit because you don't care about the organization. Just like blast it up. That's your goal,
Right.
right?
Mhm.
Everything
Mhm.
else is harder.
Right, true.
And for everything else we've done, mesenchymal stem cells, you're like squirt it there, or you squirt it here, or you squirt it there, and they sort of just distribute locally, but
Right, I see.
but it's a local application.
Mhm. Right, right. Well, thinking about brain, that's also hard to, right, introduce to a localized area, right? I mean,
Yeah, I mean, to go through the brain, if you're going into the ventricle, if you're going into
Mhm.
certain regions,
it's
it's
true,
not too bad.
right?
Where
True.
people are doing it clinically with like DNA oligonucleotides, like either through even for a sustained administration, they'll do these like intracranial injections, which blows my mind how like people are like, no, it's fine, we do all the time.
Okay, all right.
Yeah, still seems
Oh,
not
that's interesting.
ideal, but
Mhm.
But versus alternative, sometimes if you're going to get Alzheimer's versus.
That's right.
getting a-
Right,
Yeah.
well, absolutely, right,
risk
yeah,
reward.
right, right,
yeah,
right.
I think the trick is then that goes back to the duration of the response, whereas, and maybe this is again, where is the merit for doing something as complicated and maybe expensive as a cell compared to a drug? It might be only when the intervention is such
Right,
challenge, infection risk or
Right.
whatever, that it merits
right.
the need for a prolonged intervention.
Mhm. Right. I mean, one could test a lot of the things in, like, say, organoids, right?
Yeah.
Where you have, say you deliver a microglia, you could test it in an organoid, how long the response actually could be beneficial, right? You could then,
Can we pursue that a little bit? Two lines of thinking. if we
Right.
could. So, because I also believe that development of organoids or
Mhm.
something for aging research.
right,
What does that mean in practice? So do the organoids have to get old? Do
yes,
they, how old?
Right.
What, do we have the right markers? What are your thoughts on that?
right. Well, I mean, but at the Buck, we had worked with initial organoids that have different cell types, brain cell types, right? Like, and they were all iPSC derived ApoE alleles, right? So they had relevance in the context of Alzheimer's. We had only grown them 8 weeks, but then other people would say that's not long enough, they're not quite mature yet. Yeah, so maybe half a year, but we grew them 8 weeks just to as a proof of principle. How does it look? Does it have a good cell type distribution? And that all looked good. And then, as you know, so one could do some testing in like shorter cultures of these organoids. And then once one thinks that's confident and working, then one would do some experiments that are longer where the organoids can get really mature.
What limits the life of an organoid? I
Mm.
know in other systems you get like a necrotic
Mm,
core.
Right.
that, like for transport reasons, then drives this kind of degeneration eventually. Is that true for other organoids?
Well, you know, I'm the chemist. I mean, I don't know for sure, but I think necrosis is always something to look for and assess.
I worked with microfluidic devices for a bit, and sometimes one of my fibroblast lines used to just like overgrow the
Yeah.
device. And you would lose structure, you would lose stability. Somebody would contaminate their plate and it would wipe out the entire incubator.
Yeah.
Oh, yeah, I mean, that's they are like usually very extremely, don't,
I would see a
you know.
significant increase in just like dead cells in the system as well. And if you can't clear those out, then actually maybe that promotes some
That's a good
aging.
model, yeah.
And those
Right.
ES cell cultures, you can keep them going for a very long time when you're maintaining pluripotency. I was just thinking
Mhm.
like, we're talking about starting the clock, right?
Mhm.
Like,
Mhm, mhm, right.
what is it necessary to start the aging clock? You know,
Well, I mean, we talked in the other session, then maybe, you know, maybe you just have to then induce some kind of stress that makes an accelerated aging kind of happen, which could either be, I mean, some say treatment to make something senescent or, you know, yeah, maybe one has to accelerate it a little bit at some point, you know.
So that was a general feature that we talked about too in these engineered systems.
Mm,
Is how do you
right.
create a sensitized
Right,
thing? So
right.
if we, at some point, these things will work hand in glove, right? So if we start knowing what aging things we're trying to cure,
Right.
then we can pre-engineer that state. You talked about the
Right.
ApoE, so you
Right.
could have
Right.
whatever disruptive protein, or you could knock out proteostasis, or you
Right.
could preload a bunch of plaques or whatever, and
Right.
then you're pre-sensitizing to an age
Right.
state rather than aging the organoid. That seems less likely because it's never going to be 90 years old.
Right.
And so
That makes
that
sense.
seems like
Right.
a nice kind of compromise.
Yeah, yeah. Mhm.
on this topic, I don't want to divert it, but I have an idea for how like a SynBio approach that
Oh, yeah.
could be maybe pivoted to this.
Mhm.
One thing it seems always challenged with any model system, it's like, how do you know which complexity is adequate to capture
Right.
your phenomenon, right?
Uh-huh.
Right.
And we've been building as a field gene circuits for doing things like typically making from a stem cell starting point a high yielding homogeneous product target cell type of interest. Like really want to make T cells. We really want to make like beta gamma neurons or whatever the endpoint is, right?
Mhm.
We don't typically intentionally try to use like genetic tools, genetic circuits anyway, to try to create like specific heterogeneous assemblies.
Mhm.
At least it
Mhm.
happens, but not on
Mhm.
purpose, right?
Right.
There's some nice work out of like Thomas Hilliard Vecchio's and Ron Weiss's group looking at a few factors
Mhm.
and how you can get liver organoid
Mhm.
formation. And it tends to be like you just mess with one thing and like boom, organoid, multiple tissues organization scale, right? And so that wasn't intentional,
Mhm.
but
Mhm.
you can imagine if there's something that's like a good model of aging would involve something that has to self-assemble. So you can't like just mix your two cell types and hope that they form a structure that you know works. If it needs to self-assemble into something that models aging, I don't know if anything is trying to do that in a SynBio toolkit, but it certainly seems like an opportunity for
Mhm.
like the genetics /
Right.
SynBio scape to get at. Is there any way to inform? I'm thinking, so when you don't know what complexity is needed, is there some way to just like make a bunch and see which ones end up having like some predictive value?
I mean, one could make, as you say, a bunch and see which ones release markers that we think are aging markers, right? Which ones have the best aging signatures by molecular checking afterwards, right? So yeah, one could do that, right? Because we know some aging signatures that we know usually show up, and then one could then just see which systems was kind of nice regarding that or something, right?
I was thinking like the TARDIS system has this kind of feature where like you can get to a library of like what 10,000 or something.
Mhm.
I wonder like which sounds amazing. And that's a fairly complex, genetic array that's being sampled of that diversity. What do we need to do to figure out the scope of this challenge with the like, aging?
I mean, the molecular technologies are usually not super high throughput currently, right? Like to look. for
That's what I'm
this.
wondering, like if
Right.
we had an idea of figuring out, like even not today, but like
Right.
the process, like the scope of this problem is 10 to the 12th, or the
Mhm.
scope of this problem is 10 to the 5th.
Mhm.
That would have like a very different
Mhm.
driver on how
Right.
to
Right.
tackle it, yeah.
Yeah. I mean, one other thing that I think we got to in a few talks this morning is that still aging signatures are still developing, right? And that they are complex and different from tissues to tissues. So, I mean, there's maybe not a simple aging signature, which makes it harder to have a real high throughput, I guess, right? So, but I mean, one could pick, say, an organ of choice that maybe is particularly important, either the brain or like, say, kidney failure is sometimes a problem in aging, right? And kidney has also a known level of high senescence burden, so one could or like, or ovary, right, is one of the first tissues to age, right? Like, I mean, it's like way before all other organs, right? So maybe, yeah, picking something that's maybe a little easier to
Faster.
begin with, right? But-
So if I may try to bring a couple of those things together, because I do think that there's major kind of pillars
Mhm.
of synthetic biology that could be applied. So I put that under this category of engineering discovery platforms or
Mhm.
something like that.
Mhm.
Because I think that that is a major contribution.
Right.
We really don't
Right.
have the right systems for understanding aging yet. They're too slow. Even a mouse lives a couple of years. And there's no way to address this screening and scale problem, which is what you are.
Mhm.
And that's where I think that if we can do things at scale, then we can actually select for
That's right.
the cell
Mhm.
types that are most useful or whatever. Mutate them or add things or do whatever. And then the other thing that keeps coming up is can we use these synthetic biology approaches to actually do a better job of discovering what these hallmarks or signals of aging are?
Mhm. Right.
Because that feels like that's a major limitation, because we wouldn't even know if we have the right thing, because we don't know what to measure.
Yeah,
And so what is the roadblock for measuring the right thing? Like,
Mhm.
I think if we think about that, like what problem needs to be solved in order to do that? Is it because aging is so heterogeneous, we don't know what it is? Or is it because we don't have the right tools
Mhm.
for readout, systematic, looking at the whole proteome, is that sufficient? Or we need to manipulate things, that those are some of the challenges that I think synthetic biology could help really leapfrog the field quickly.
Can I build on that with an idea? This actually dovetails on something we talked about in our last
Mhm.
session too, which is there's sort of like a question oftentimes about heterogeneity at
Mhm.
the level
Right.
of the not just between the organs or between the individuals, but even the cells within an organ,
Sure.
and trying to understand maybe why in a single organ, a single patient, some cells have one outcome and some cells have another.
Right.
We talked about the possible utility of having some ability to record
Mhm.
the states and maybe inputs that
Mhm.
different cells had received as a thing that could be a useful tool. And the bridge I was going to make was, I was imagining, are you guys familiar with the Brainbow experiments?
Yep.
They're just so beautiful, but it's just this way you can track, it's sort of a randomization of which fluorescent protein gets expressed in a cell over development, in the
Mhm.
brain in this case.
Mhm.
And then you can kind of have this, each cell ends up being a different color. And then you have like this really nice way that you can actually reconstruct the structure of all the neurons,
Right.
because they through randomization
Oh, that's cool.
have come up with different colors. So I'm thinking not the Brainbow per se, but thinking about like, maybe there is a way you can imagine like different perturbations. Like,
Right.
let's say there was like all these different things that when it turns on this gene, something goes on. We know it turns on at some point during aging. We're not sure like what makes it turn on in a way that is good or bad. So we like build a bunch of circuits, like reconnect stuff in different ways, but you do that in a way where this happens in a single animal. And then you let it age. So it
Mhm.
may take a while, but there are like 10,000 different
Right.
circuits in the animal.
That's cool.
And then you look over the course of time at which circuits
Yeah.
persist as a function of the aging process.
Mhm.
And you can
Right.
see which phenotypes are selected for over the course
Mhm.
of aging.
Right.
And
Cause of aging or even inducing stress.
something, right?
Where which cells, which organs are resilient, which ones are not, and
That's right.
you know,
Bin them into the
mhm,
ones that
right,
maybe have
right,
this kind of
right.
stress
where-
markers.
and or also if there's, say, you have an insight on or a stress, at what age can the cells overcome it, and at what age can they not anymore, right? So that could bring a lot of interesting questions.
Right.
And I think I love what you said, because I think there's something that's totally not understood. There's the stochasticity in aging, where even when things supposedly are the same, some things happen to one cell,
Right.
not the others, or even on the animal level, say like some model animals, some die early and others not, even though they all have the exact same environment and
Right.
genetic code. So there's a lot not understood that maybe with such a monitoring system could be informed.
It could be that. And I guess I was also thinking just in general, the challenge with organisms with models is that either they like take a long time
Right.
or they're expensive
Right.
or whatever. And so one way, I'm just thinking of models, another field that I'll mention here. So like for people who are interested in the banana particles
Mhm.
and they want to figure out how to make the lipid composition cause delivery to go to a given place.
Mhm.
It's generally agreed upon that like nothing predicts what's going to really happen except people. And the closest you can get is non-human primates. And that's like $1
Oh.
million an experiment.
Mhm.
And so it's really expensive, live a long time. And so they use these barcoding approaches where you like link chemical variation with the DNA barcode.
Mm.
And then in a single animal, you inject like 10,000 different formulations.
Mm.
And you're assuming they're independent. That's one limitation, which is also an assumption what I said. But then you just do this and then you wait and then you look at the target tissue and you sequence the DNA and then you back that out to
Mm.
which chemical composition gave you that. You can sort of manage this issue of the animal model being
Mm.
limiting in cost or time. I'm wondering if there's like a way to do that kind of multiplexing barcoding for all these kind of things we're talking about, like to deal with the fact that we don't know what to measure, right? Is there a way to make use of, we can simplify, and I think make it faster is one thing, but when it has to be in the full complex model, are there opportunities to mitigate those costs?
I lost track of the chicken and egg in your, where you're going. Is this
I-
to
I-
identify, is this to identify potential therapeutic interventions or things that are drivers, or is it to try and measure what's going wrong?
I was thinking of it more as a discovery tool in this
Uh-huh.
way and thinking about like, just in general, if we're not sure what to measure and maybe there's a way that we could use some kind of interventions to help us understand what are the drivers of these processes or what are the causes of these outcomes? How do we manage that limitation when the model has to be big or slow or something?
So I like the idea, and that's how I think all the time. The issue is, what's at the end, what does success look like when you're gonna just say this
Yeah.
worked or it didn't, or
Yeah,
it was informative or it wasn't, so.
my
Is it
naive,
the cell is alive? Is it, whereas if you had a reporter that you were looking for, then you could sort cells based on that or.
yeah,
something
my
like that.
proposal was more like the latter in the sense that like if you had stem cells that over the course of time had transitioned to a more aged state, whether it's senescent or otherwise, and others that did not, you could then bin them that way.
Yeah.
Look at those circuits or those interventions.
So
and
there
start-
is some hallmark of aging that you are defining as an outcome. That's basically where I was
At
going.
least the way I was
So
thinking.
you have
about
to
it.
pre-know something about it.
Something,
So-
Mhm.
No, I agree. I think that that's good and you
I
can certainly
guess-
do that.
It could be that, or it could be. I guess this is not, this is confusing me now, because I was thinking the cells that die really didn't age well. So this is like aging versus aging well. Like maybe when we look at the mouse,
Maybe you want
And-
to see the dead cells.
what's
I
that?
said, maybe you want to see the dead cells.
Yeah, maybe you want to know what the dead cells were.
But maybe they
right?
did age well
What did they
because
do?
they killed themselves appropriately.
Oh,
Yeah, yeah, yeah.
it became detrimental, yeah.
Those are just different interpretations of
Yeah.
the same data set.
Yeah, exactly.
Yeah. Huh.
I think, I mean, what we would want are things that maintain function late in life.
Mhm.
And so however we're gonna measure that.
Yeah.
It could be that they're still around. It could be that they're
Mhm.
able to be sensitive to insulin or
Mhm.
to a neurotransmitter.
Mhm.
Or
Yeah.
whatever the thing is, that there are readouts for that. I think that last thing that I just said, because there's a lot of synthetic biology in neurobiology
Mm.
that is not
So much.
it's not the circuit-based way of thinking. It's again, tools-based, like how can you tell that calcium has occurred and you know,
Mm.
and then image that. So
Mm.
there's ways of probing
Function. I love that because I think that's sometimes the problem, right? You don't know, is this tissue piece the functional, right? Like, or is there a degree of function, better function versus not good function, right?
That's a good point. And
Yeah.
you could take a lot of bins from, because neuroscientists, I was actually struck by neuroscience is like richly steeped in the circuit concepts and like how all that works.
Yeah.
Great ways of studying circuit function at various different levels of genetics, not the molecular circuits that
Mhm.
we're talking about.
Right, right,
That could be a good way of thinking
Yeah.
about like how to ascertain
right.
function and dysfunction.
Right, because sometimes it's interesting when you come from I mean, I do a lot of molecular measurements. We know a lot of what the molecular landscape is, but we don't know much about function, right?
Mhm.
Bringing function to data sets, that would be really cool, right? Like, because I mean, aging is so difficult, right? Because I mean, aging by itself may not be so bad, but what is really bad is when it loses function, right?
Mhm.
And then the question is the functional decline. How is this? How is it, right? Is it like it just gradually goes down, or is it like doop, and then it falls down, or you know? So one could learn.
Yeah.
about
So
that.
just from our own work, I've thought about, can we have a readout of neuronal function
Mm.
late in life? Challenge thing so that it turns on, whatever, and then do a mutant screen on that. So that you can
Oh
start
yeah,
combining scales of interaction. Ultimately, I do think getting this into some sort of human tissue approach is gonna, for this general class of problems around aging, is gonna be relevant. But I just think they're not addressing things at the appropriate scale. And that's partially because we don't have a good phenotype that we can select on.
right.
In order to do a variety of things. So the
Right.
more, that's another application of the discovery platform, like can synthetic biology give us a nice phenotypic proxy that then is screenable
It's a
or
great idea.
measurable.
Mhm. Right.
There's so many good examples of how like one of those transforms the field, right?
Yeah.
These
Right.
like transgenic mice just transformed T cell immunology.
Uh-huh.
One,
Right.
if you, it's like the one couple of strains that are like the OT1, OT2s.
Mhm.
It's the whole like millions of papers on ovalbumin responses
Yeah.
because it
Right.
lets you do that, right? It lets you like actually
Right.
parse it out.
Right.
I wonder if there is what would that look like? What would that like to have we have an aging marker or like late age, middle age, whatever. What would that ideal spectrum of tools look like to then provide a basis for screening?
Well, I think, you go ahead.
Thanks. So I was wondering, as the epigenetic side of the aging, one of the discovery was the DNA methylation kind of correlates with the aging.
Mhm.
I think there was the Horvath clock.
Mm.
I brought it up in the last session as well. In the embryonic stem cells, they are basically non-existent. But as the cells are passaged in vitro, the amount of methylation increased. And I'm pretty certain there have been recent articles about across many different organisms, such pattern was conserved. So I was wondering if that can be somehow we can take advantage of that information to, I don't think it's a phenotype per se, because it's
Right.
DNA methylation.
Mhm.
But it's a biomarker, I should say.
But you could have, that's what I would take that circle. And then try and get a thing per slice, right? So that's epigenetic state, so.
Yeah.
This is the circle from your slide.
Yeah,
I wrote
so
that
then
down,
there's maybe you're not surveying. the
That's right.
Higher epigenetic state, but you have a reporter
Right.
that then turns
Right.
on when its epigenome
Mm.
gets modified. I think that there could be.
Yes.
pH related proteostasis, sentinels in there.
Mhm,
We just talked about, being responsive, to some sort of, neurotransmitter
Mhm.
challenge.
mhm,
That's where you could go for this brain organoid thing.
right,
It's like, what is
Right.
an aging thing?
Right.
It stops lighting up under serotonin,
right.
something like that. And what's the
Mm.
rate of decline there?
Mhm.
And then what helps maintain that? So
Mhm,
I think you could choose,
right,
potentially macro functional
Mm, right.
components, rather than the micro functional, like, does this protective
Right,
protein
right.
occur? But what's a physiological, I guess that's what I mean by
And-
macros, like what's an
Right.
integrated physiological?
And actually that's,
Yeah, integrative and maybe.
right, I like that. Go ahead.
I was just gonna say, I think the trick is like when things we can think of most easily are like, well, we know that when this gene turns on, it's important. And we
Yeah.
can sort of like, even if that doesn't exist today, we sort of know how to do that, right? But we don't know how to do these integrated physiological
right.
reporters, right?
Right,
Like,
right.
when does the whole state achieve this like
Right.
poly variable
Mhm, Yeah.
multivariable
right.
determinant, yeah.
Right. Well, and I mean, I think that's really so difficult to assess in a living organism, a functional readout, right?
Mhm.
And that would bring value, right?
Mhm.
That understanding and,
Yeah,
you know, yeah, I think that's an interesting thought.
are there any big gaps on this list? Like when we talk about, so senescence, we've talked a lot about mitochondrial dysfunction is when I keep bringing it up because I know least about what that would look like.
Right.
I know like you can look at like turnover or something, but are
That
there
could be,
other?
but also energy production, right?
Yeah.
Like, so for example, electron transport chain complexes may not work as well. Also, the mitochondria in general have a lot of energy production. So that's why, for example, also, so NAD treatments, right? They work, they are energy kind of little contributors, right? Like, so, yeah, I think, I mean, that could have functional readouts potentially also.
Is there a
right.
way to like, if you wanted to know in an animal how the mitochondria were doing in all the cells
Mhm,
over time?
mhm.
Is that doable?
Well, no, well, if you
Way?
take them out, then you can measure energetically, you know, but in the living animal, that would be more interesting, right?
Mhm.
Like,
But there might be some ROS.
Right.
Yep.
Sensitive.
Right.
Readouts.
Yep.
And that's
Right.
where, again, from a synthetic biology point of view, I think the circuit-based thinking that you need amplification of signal. So you might have something that's sensitive to ROS, but are you gonna be able to see it? So you need to couple that to something
Right.
where
easier to.
you have
yeah.
like, if you could define, this is a cellular readout thing, that's GFP or whatever.
Yeah.
And then if you could attach different
Right.
things at the front, and then
Yep.
they go through an
Right.
amplification cycle.
Those totally work. 'cause
Right.
You can do things like background subtraction and you
Right,
can do
that's
things
nice.
like level setting and you can convert a small, because like most gene expression changes in real life go from like 1 to 2. That's the physiological range
Yeah.
of like meaning. And so you just can't measure that in anything. So this idea that you can then convert that like small analog range to like a 10-fold digital.
Yeah, that's
switch
nice.
It's
Yeah.
required to be able to actually
Right.
monitor it.
Yeah.
That's well within the range of
Right.
what.
Right.
So that literal thing, we've done that the first time we tried, which was
Right.
just to take a systems biology principle, map it into these little pieces, and you can do these kind of analog to digital converters.
Mm.
And so if it worked the first time, it must be doable for others. It
Mm,
just
cool.
wasn't that
Right.
good of an idea.
This amplification in general is somewhat interesting to me. Because sometimes biological signals are a little bit more mellow or diluted
Yeah.
by a lot of other cells, right? Particularly say if some cells age earlier or, you know, like senescence burden is very low, abundant amplification of signal seems interesting because then you could see things early on and then see as it progresses. Mhm. Right. Yeah. I mean,
Tempted, I just wanted to use that one as an example. Like, I wonder how many of these other topics, there is something that's kind of like a gap in how we,
right.
that discovering what's going on seems like a fairly big win at the end of this.
Oh, for sure.
There are
Right.
like intermediate wins along the way that are more like, this would be a great tool. I don't know what I'm gonna find, but I like can't study
Right.
this. Right.
I mean, I think, and I mean, we talked about that in the other session, the dynamic aspects of aging, I think are not very well understood in vivo, because when you do anything animal or human, you have very distinct time points.
Right.
But I mean, what is interesting is the dynamic nature of aging and trajectories. And because that would also be important to know where and when are times of best interventions.
Right.
Right, like, and or how long you do some treatment, you know, and right.
Can I ask this? I've been just mulling this. I'm just gonna say it out loud. This idea of like including a functional interrogation is really interesting. And I'm wondering if there's something we could do with that at the level of, let's say, patient materials. And here's what I'm thinking. There's a very, if you look at like neurons from stem cells from patients who have different backgrounds, some of which are predisposed to neurological disease and some which don't. If you look at markers in compositions, they look identical.
Oh.
But if you then start like trying to stimulate and interrogate the responses, suddenly all this stuff becomes illuminated, right? And similarly, when I was a postdoc, actually, we were looking at this kind of monocyte phenotype. And when you looked at the monocytes from all these different patients, they were identical, they had the same things, the same things more or less, but there were certain types of stimuli where suddenly you get bifurcation of the population. And it was a function of like the local immune state of those patients, because it would change as a function of the donations for the same person over time. I'm just thinking that like that idea of like functional interrogation of patient samples.
Right.
I'm thinking those are easy things, right?
Right.
This is like dump a cytokine on or a TLR ligand, see what markers turn on. You can conceivably do more sophisticated versions of that to like interrogate
Right.
patient's tissue over
Right.
time to like define aging,
Right.
right? Or
I
biological.
find that also super interesting, because I also think that when often, you know, a tissue may be totally fine for months and months, but if it had gotten some insult 3 months ago, it would have not done well in that, right? And so the reaction to stimuli, that gives you also insight into, is there still resilience, or is this aging process? Because you could imagine, say, two people of the same age, and then one person gets a stimuli, nothing happens, they're totally cool, and the other tissue starts to have problems, right? So having a readout like that, I'm sure, would be super interesting, because you would know before it would happen in the native tissue, right? So yeah.
So I'm gonna call that a resilience challenge.
I like that.
Yeah, I was thinking like this idea, like the liver might be doing fine, but maybe
Right.
if you had a liver biopsy and you could do a resilience challenge, you could predict whether that patient was at risk
Right.
of.
Right.
The thing that just popped in my head is, you know, some of us are old enough now that we actually have to do like a resting metabolic thing.
Sure, yeah.
And then you have this insulin challenge where you take a lot of sweet stuff, and it's the same kind of.
totally.
Right, exactly,
same kind
right.
of thing.
Or if you have
Because
an
normally
EKG
you're
after
doing
stress,
fine,
Yeah, same thing.
right? Yeah, right. Normally you're doing fine, but then if you get all the sugar, then it's like, oh no, right? Yeah, I agree. That's
And I guess this is very much kind of a medicine systems biology thing. I don't know if there's an opportunity there for SynBio to do something. Circuit, but I mean, like, I don't know if there's a,
Well, it's
if
the
there's a-
it's the readouts, again.
oh, the readouts, yeah, that's a good point.
I think
Right.
are, you know, what would we look at? I thought I had this in here.
What time scales are important for these readouts?
Hmm.
Like if you're looking at some type of brain process, it's gonna be very, very quick. That's gonna limit what synthetic biology tools can be used
Hmm.
for.
I see.
So what type of timescales would be important?
sure.
I think they could be. I mean, we're talking about physiological readouts of the current state of the cell, so I think that they would in general be kind of fast-ish under a challenge, so even metabolically or, you know, there's static stress or these different things, I think. As opposed to necessarily needing to engage the whole. I mean, it might be involved gene expression, but maybe not cytoskeleton and that kind of, that's
Yeah.
slow.
But maybe like I'm thinking the calcium gated reporters, right? You have this kind of like fast conformational change that tells you whether calcium fluxes happen.
But there must be something like that that would respond to, you know, a redox state
Yeah.
in the cell.
Yeah.
That you could give a bunch of energy or glucose or something, and then all of a sudden the thing turn on the way that you wanted it to.
Yeah, it's neat. And the more you make more than one of these, you start needing some tools to actually like deploy them in a way that's like meaningful and calibrated and
Mhm.
constant to be able to have your,
Mm.
you know, interpretation.
Mhm.
Just wondering, just going back a few conversations, you mentioned about two identical looking cells. When put under the resilience test or stress test or stimuli, they behave differently.
Mhm.
So when you say identical, like what kind of aspect are you referring to?
I don't know.
Okay.
I literally don't. I was thinking naively, it would be like they're from the same tissue as a
Uh-huh.
starting point, same patient, same point in time.
So maybe same cell type then?
Yeah, so just in
Uh,
general, like if there's something where like for whatever your characterizations are, it seems like they're the same, but they have different outcomes and you want to know why.
I see.
So like even if they're starting with the same phenotypic
Mhm.
information, you can have macrophages in one tissue and have different subsets of macrophages within
Mhm.
that tissue as well. So
Mhm.
if you have two that for most of what we measure, they look the same, but functionally they are different.
It can be that, and I think the complementary scenario is there is no underlying difference, but there's like a symmetry breaking that
Right.
occurs, which can be stochastic.
Mhm.
I see.
Both are probably
Yeah.
happening, yeah, so.
Can I do a quick check? And so I'm dividing the world in because this is, how I think about it already, but let me know. Between these kind of discovery platforms and
Right,
therapeutics.
right.
We've talked a little bit about therapeutics, but not a lot, but I think like, ooh, what kind of cool therapeutic. I think it's way early for that.
Yeah, right.
But there is this in-between ground, which are discovery. And is there, what does that space look like in this synthetic biology realm? Like, can it help us discover new drugs? Can it help us make new drugs? Like what's going on with that?
Yeah, I mean, as far as like. We have talked about several things that could be a path to translation, that these fundamental discovery tools could be mapped. I think not all of them yet are clear, but drugs is one. I think we talked about cell therapies as a translationally relevant thing that could come out. That's not specific, so that covers a lot of ground for both those things. Are you thinking about what other things might go into that box?
No, I think just like, for instance, if you had these readouts, you could then screen for drugs.
Yeah.
Mhm,
The
right,
readout
true.
systems. which
Oh,
one-
does it make this better, right?
Yeah, which one? But then you,
Which drugs actually work.
Right,
exactly.
right,
So, but
right.
then you would like, if you could have these heterogeneous, we're just combining everything together. We have these heterogeneous organoid systems that we then engineered with the right readouts for neurons. And we could raise 1 million of those independently and
Yeah.
then
Right.
test 100,000 compounds on them.
right. That's
Then we're
super
getting
strong.
somewhere
Right.
about where
Yeah.
we need to go rather than. We're just very precious in all the work that's done right now. I think we just need a systematic thing
Mhm.
for some of these discovery parts, and then on the genetic side as well.
And
Or do we engineer sensitized systems that then we're curing with a drug, and then we have the right ways of measuring?
Is there a similar challenge? I was just thinking if there's an analogy here, when we did one of these synthetic biology and cancer workshops, one of the big illuminating insights from the clinical oncologist was like, guess what we don't need? More drugs.
Yeah.
The reason is we don't know how to administer what we have in the right combinations.
But the problem is, though, in aging, there's just nothing.
That's what I wanted to ask.
I
you,
mean, the
so-
thing is, for cancer, there's millions of drugs that, because there's a huge industry that has pharmaceutical industry that has done this for decades.
Yeah.
But in aging, for example, the clinical trials that I showed, there's very limited
Well,
things,
that's what I
right?
was wondering, and would it
Right.
be also the case that even with those limited, like, interventions, combinatorial, understanding like dose scheduling
Absolutely.
and combinations? is hard?
Yes. Oh, yeah. I mean, I have, for example, even this D and Q, right? Those two senolytics, it's known that they together work much better than individual ones. So the whole combination therapies, all of this is really not as well explored, particularly also not. I mean, if you think about, you could think if one had such a screening technology platform, you could screen this for different tissues and organs, right? Like, I think,
That's right. You
I
can
think-
shift where those
Right.
molecules get delivered.
right, exactly,
how,
right. Yeah.
whatever.
I think that's very different from the cancer field where there has been so much effort forever to define drugs. And
But I think
this,
it was in
right.
that bin back in
Mhm.
the early
Right.
chemotherapy combo
Right,
trials, right? It was
right.
like, what if we just like
right, right,
give them everything?
right.
Yeah.
But
I
you
mean,
have platforms for.
Right.
Leah Croucher
There's-
also wanted to say that synthetic bio is used to produce hard to synthesize natural products with therapeutic effects. Sorry, that might have been a couple of beats back.
Yeah, no, that's right on target. Go
That's
ahead.
right.
Nisha.
Yeah, I was just gonna point out, we had
Oh,
the
that's
chat.
what you're pointing out.
Oh, I
Okay.
see, sure. Well, I mean, I said in my talk earlier, I've been a big believer of natural products as ground structure. And then manipulating that to find whatever works best for the system, right?
Mhm.
So if that's a feature also, that could also be super interesting. I mean, the problem is really that the molecular readouts are, I mean, they're great, but they're also very expensive, right? Like but I mean, if one had a screening tool that could do so much, that would be quite valuable, I think, right? Mhm. Right, that's cool, actually.
Just a random thought, as a very layman perspective of it, because we are originally talking about the phenotypes of aging and all that. It's kind of very odd to just think very back, like a very average layman perspective. That I can tell, at least I think, it's like by looking at people or even animals, like how old they are. But then again, when we want to look at the cellular
Mhm.
level, that becomes like extremely hard to get what are the phenotypes,
Right.
biomarkers to
Right.
really detect what they are. It's kind of a very odd kind of a what is it? Phenomenon there. Is it a emerging property as we go on a higher level of
Well-
organs or people?
Mhm. That's an actually really interesting point you bring up. I do think that there are some aging markers that show up all the time. Some are all the time, like GDF15, periostin, a lot of ECM markers. We talked about this earlier. It's just that I mean, it's kind of funny because a lot of people do RNA sequencing transcriptomics, and a lot of the phenotypic markers that happen in the tissues. Well, I mean, if you look at a transcript, that's not necessarily what you have later on a protein level. So I do think that there's actually value looking also more at protein levels. And so I think maybe that will change at some point, where on the molecular level, we will also find more things that are really robust. But I mean, I think we're maybe just starting, right? Like,
Yeah.
I mean, but it's an interesting point you bring up. This is
So like ECM is natural, and how we define it as a dysregulated state
mhm,
is often context dependent on the person looking at it.
mhm.
I would have a couple of responses. First of all, you're exquisitely evolved to pay attention to people's faces.
Yeah, right,
Mm.
And so a lot of your
Right.
assessment of age
Right.
is based on
Right.
that.
right.
So whether or not you could predict how fast they could run based on how old you look at them, or whether or not they were diabetic or these other than other physical or whatever, these other things. I don't know. So there's a chronological age and the functional age of any of these tissues. But I do think you're correct. Anecdotally, and this has been quite a surprise to me over the last 10 years. The signal that we see again and again in a lot of these aging studies is collagen.
Yeah, right.
And so that is a
Right.
major feature of what makes our skin look old.
Right,
And I do think that the intercellular matrix and all of those kinds of things, and that is another feature that's kind of entrenched if you think
Right.
about it. So you kind of lay that stuff down,
Right.
cells are replaced within
Right.
it. Are they restoring it? And that I think we don't really know a lot about. And that is a strong correlate with the way we look.
right.
And so, is that important, in our heart tissue and our face? Are those the same features that are drawing it? I don't know, but.
I'm not suggesting we prioritize that, but if you fix that, we'd all be rich.
No, that's correct, yeah.
But I have I agree with you. So I have two thoughts. One is a little funny. I think Josh can run faster than I. We could test that, no, but I mean, I think that's really cool what you said, we are so trained in looking at facial expressions or statue. But can we train algorithms also to do that, right? Like, for example, at the back, we had done a little bit some retina scan work with Google together, Pankaj Kapahi, where that then was a really good readout for obesity or something, right? So the question is also, can we have creative thinking of, out of the box things along those lines, right?
I was trying to think about how to work that in. It feels like there's all this opportunity for like behavioral modification, where like a lot of
Right.
times behavior is modified after the crisis has occurred. If you can figure out a way to like give people information ahead of time that would
Right.
drive a behavioral change, this is
Mhm.
the synthetic biology.
Well, that's right. So some of these things are, but like what you were saying, these are bioengineering
Right,
problems.
right.
Like how do you create a machine learning?
Right.
Or assess the
Sure,
whole
right,
cellular state, but they're not synthetic biology.
right, I
problems
know.
per se.
right.
I was just gonna say, I don't think you've had to deal with an 85-year-old and trying to get them to stop having dessert at every meal.
Oh, I think at the point, they're like, what's the worst that can happen? That's what I'm saying. But, like, if you were like 35 and you realized that there was like a substantial chance that if you
Right.
made a minor change
Right.
in your lifestyle,
Right.
it would have a change.
Significant impact.
Yeah, this is sort of like the diagnostic risk benefit analysis, right?
Right.
Are there aging
Right.
related like risk benefit analyses that could
Right.
be personalized, but this.
There are, and we already know them.
Yeah, I know.
We have obesity epidemic in the country. We have all these, those are all.
Yeah, I
uh,
understand. That's true.
That's why
Right.
longevity is going down in the United States right now. So.
Mhm.
Yeah,
Yeah,
Also
that's true.
COVID, right? Did a
Yeah.
little contribute to that? COVID?
it did.
Right,
That is-
right,
that is
right.
also true.
True. But I mean, this is actually in general, I mean, just a general comment. I feel aging needs to have more educational components towards lifestyle change, I feel, right? Like, because what you just said is like, yeah, if some people would make some interventions themselves, they could change a lot of things,
Yeah.
right?
Well, I guess I'm also thinking like there's a moving target to some of this too, like, have you guys read A Natural History of the Future?
Mm.
It's a really good book, but it's one of the precepts is that as the climate warms, people are going to be subjected to different types of heat stress, even if like no matter where you are in the world,
Yeah.
that's going to be true.
Right.
And so that's like a different stressor that we
Right.
do not have like cultural,
Right.
social adaptation to
Right,
in
right,
a regional specific
Right.
manner. We do have cultural adaptations that differ across the planet.
sure,
I,
right,
I was just thinking, is there a way to layer in a biological component to some of these like new
Mhm.
stresses?
right,
That's really what I was thinking. It's not really different from the diagnostic that you were talking. about.
right.
Maybe it's too far afield. For this
Well, I think
Interesting,
creating
though.
a tighter feedback loop. So
That's
if we
right.
are able to
Like a
do
more
something and then you ate a dessert and then a little button went, you just took 10 minutes off your life or something. That would be.
I feel like at that point, I'd be like, that's okay. Closing out early.
Oh, in 25 seconds, okay.
I think it's supposed to go to 3:25, so maybe it was just programmed.
5 more minutes.
I'd say
Yeah, 25, right? Yeah,
Yeah, 25 on
Right.
the.
we won't be able to keep
thing.
recording, but we can keep discussing.
yeah.
Okay.
But it's, yeah, it's just thinking of these kind of like novel stressors. One reason for the devastation of the COVID pandemic was like, we didn't know